Hello friends and followers, welcome back to my channel Microscopic Biology. Before we begin, please like, subscribe, and share all my videos. Today's video topic was requested by Chemistry Talk with Dan. So, let's get to it. So before we look at the Angiosperms, Gymnosperms, and Ferns under the microscope, let's learn a little more about what these classifications are and characteristics of these classifications. First of all, we have Angiosperms. There are flowering plants that bear their seeds in enclosed clumps, whether it be a fruit or a seed pod. This group is comprised of almost all the plants we know. Some examples are fruits and vegetables that we eat, and trees like oak, hickory, maple, and pecan. The next group is Gymnosperms. The term gymnosperm is Greek for naked seed. This reflects on the unenclosed condition of the seeds that they produce. Some examples include conifers like pine and fir and cycads. Lastly, but definitely not least, we have ferns. This group is seedless. Instead of producing seeds, they reproduce by spores, which can easily become airborne. These spores are produced by sori on the underside of the pinna or leaves. So for today's experiment, I will be using the Angiosperm, Nandina domestica, commonly known as firepower. For a Gymnosperm, I'll be using Pseudotsuga menziesii, commonly known as Douglas fir. Finally, I'll be using a Nephrolepis exaltata, which is commonly known as Boston fir. With that being said, let's hop into the microscope. So first off, we have the Angiosperm Nandina domestica, which is known as firepower. And as you can see, angiosperms have lots of stomatas. The stomatas are those black dots in the oval shape cells, and stomatas allow for the exit of carbon dioxide, and some moisture passes through, so it maintains balance within the layers of cells in the leaf. Here we have a small vein. This vein actually branches off from a larger vein, and this vein provides nutrients for the smaller cells around it. Here, as you can see, is the larger vein. Take note of the fact that the vein has large rectangular cells, whereas the rest of the leaf had circular cells. This reason is because transport of nutrients and water that goes through these cells occurs quicker on rectangular cells and longer cells versus shorter spherical shaped cells. Next up, we have the Gymnosperm, and this is a Douglas fir needle. As you can see, that vein that runs down the center of the needle is the only vein that is present in conifer needles. Conifers only have one vein, and that's the reason that they are needles and not broadleaf. Their leaves are not wide, so they don't need any side veins cutting through. So as you can see here, this is a cross section of the Douglas fir needle, and the ridges of the cells all neatly stacked on top of each other can be seen here, and this allows nutrients and water to be transported very quickly in between them because there's only one vein to distribute water among the whole needle, and that vein, as you can see, is clearly outlined on the right side. And if you look in the middle, there aren't any other veins to be around because the needle is very thin, and that helps the plant to conserve water, helps it to conserve moisture, and maximize the the photosynthesis output, which helps these plants to be evergreens. Lastly, but definitely not least, we have the Boston fern right here, and as you can see, that is a sori. That is where all the spores are produced, and those spores, they become airborne. They settle in places
and then grow to become new fern plants. As you can see, that vein that goes into the sori connects to another one of the spories at a Y-shaped intersection, which you can see right there. And that Y-shaped intersection later goes into the main vein, and that main vein is actually tinted a slight red, and that is because the fertilizer that we use for this plant was actually tinted red. So as you can see, the capillary action did take up that red dye with it, and thus it did dye that vein a slight red.
Thank you so much for coming along with me into the microscope to see these wonderful angiosperms, gymnosperms, and ferns. The idea for this video was brought to you by Chemistry Talk with Dan. If you haven't already done so, please visit his channel, hit the subscribe button. But before you do, please subscribe to my channel and hit that like button. And watch all my other videos. As always, I'm Mike from Microscopic Biology.